everyone. I'm Meng. I'm an R&D scientist working at Vancouver Coastal Health. Today, I'll be sharing some insights from our lab's quality improvement projects focused on lab-developed LC-MS assays. Specifically, we will discuss the importance of internal standards and their role in achieving accurate and reliable results in clinical tests. And I'll also present two real-world troubleshooting cases where internal standards selection actually enhanced our assay performance. We'll also get into some technical aspects of LC-MS, and it can get detailed, but I hope that this will highlight the practical considerations for using internal standards in clinical applications. Let's get started. So first of all, some background information about LC-MS. Mass spec is now widely used in clinical lab and clinical research thanks to its high sensitivity, high specificity, and the flexibility in customization and multiplex. It is suitable for measuring a wide range of analytes, including small molecules, hormones, vitamins, drugs, and some peptides and proteins. In today's talk, I will be focusing on specifically on small molecules measurement, as peptide and protein are a very different story. And a typical workflow for lab-developed LC-MS assay is shown here. So in the sample preparation step, it extracts the target analyte and clean up the sample matrix using techniques such as dilution, protein precipitation, solid phase extraction, or like supported liquid extraction, etc. I'm not gonna get into details of those techniques. And then after the sample are extracted from the sample preparation step, they are injected onto the liquid chromatography separation system. So the compounds on the liquid chromatography separation, and they are separated in sample matrix based on their affinity to the stationary phase column material and the mobile phase solvent. Typically, this would be a reverse phase column. Polar compounds will usually elute first, and less polar compounds will elute later. Eluted out from the LC, the target analyte, these like red beads here will get into the mass spec. Here I'm showing a triple quad mass spectrometer as an example. The liquid phase sample will first get ionized. Basically, sample gets evaporated, and the analytes together with the sample components will form gas phase charged ions. And these ions enter a vacuum space that filters target ion with preset mass to charge ratios in this first quadrupole. And then these ions will get fragmented in the collision cell. And then the last, the target product ions are filtered in the third quadrupole, Q3, and get detected. The precursor ion and the product ion together is called a transition. Each molecule, based on their chemical structure, they will usually have their signature transitions in a given mass spec condition. And by monitoring several of these transitions, we could monitor a specific compound of interest. And then in each batch, we will run calibrators with known component, with known concentrations, along with the unknown patient samples. And the entire batch is processed exact the same way and inject to LC-MS. Then a calibration curve will be generated based on the mass spec response and their known concentrations. And this can be used for us to quantify the unknown samples. And then despite using the sample processing method that's exactly the same, the recovery variances can still occur across the batch. In sample preparation steps, also LC injections, plus also mass spec ionization efficiency could be different. On the other hand, the matrix of each patient samples also varies, which may lead to a slightly different matrix effect for samples across the batch. It is strongly recommended to add internal standard to normalize the variations in the batch. So here we add an equal amount of internal standard to each vial in this batch, including the calibrators, unknowns, and QCs, so that each vial, theoretically, they should show a consistent internal standard mass spec response. And we use the ratio of the analyte response and the internal standard response to generate the cal curve. This could help correct for the variances and normalize the mass spec matrix effect across the batch. Then what makes a good internal standard? Here are some recommendations I listed over here. First of all, we would want the internal standard to be added to the sample as early as possible. And this internal standard has similar physical chemical properties to the analyte, so it would mimic the analyte in the entire analytical process, including extraction, LC, and ionization in the mass spec. And it's recommended to use a stable isotope-labeled version of the analyte, as this internal standard would closely mimic the analyte's physical chemical properties. And in order to make it easy for the mass spec to distinguish between the analyte and the internal standard, the mass to charge ratio difference between the internal standard and the analyte is usually needs to be at least equal to or above 3. Also, we would need the internal standard to be chemically stable. So we should avoid the deuterium exchange and deuterium hydrogen exchange at those exchangeable positions. And then we also need to confirm there is no endogenous compound interfere with internal standard. Also, we wanted to choose a vendor that can provide a consistent supply for method development and routine testing. Okay, after talking about the general recommendations for internal standard, let's look at 2 real-world troubleshooting cases in our lab. The first case here is urine 5 HIAA test. Our lab has used this assay for nearly a decade with a very straightforward dilute and shoot sample preparation method. So 10 microliter of urine is diluted with 200 microliter of dilution buffer, and we add 25 microliter of internal standard, mix, and inject to LCMS. The LC condition is a very typical C18 column on a UHPLC with a 0.5 ml per minute methanol water gradient condition, also very typical, and then to inject it to a triple quad mass spec. And here is some history and context for our HIA assay. So first version, before introducing to the mass spec, we originally used HPLC with electrochemical detection, and the first version of a mass spec method used the same single point calibrator as the previous HPLC method, and we introduced a D5 deuterated HIAA internal standard. And then due to supply issue in the second version, we switched to a D6 internal standard and validated this method changes. And then the single point calibrator was discontinued. We then moved to another vendor 6-point calibrator in version 3 of our method. And this method has been run for years and very robust and never really had any issue. Until recently, we began noticing some cal curve issues in our HIA assay. In the left is an example of what we typically see, a very nice calibration curve with R-square of 0.999, which is pretty good. But on the right is cal curve from a recent batch where we see a R-square being only 0.993. And the calibrator 3 and 4 deviates by 20-25%. Seems like there is a linearity issue. And we also tested 2 proficiency sample, and with the result still being in the allowable range, but slightly positively biased. It looks like we the Cal6 linearity issue is causing this issue. So we removed Cal6 from the calibration curve. We saw that the curve linearity improved with a very good R-square, and the deviation is okay for Cal 1 to 5. And the proficiency test sample result falls back into a more expected range. But this left us wondering, is there just a Cal6 that's a random outlier, or is there a sign of a deeper problem, or is there truly a linearity issue in our assay? At this point, we started the troubleshooting process. I am just going to show how we approached this issue, and there could be a lot of other ways for troubleshooting in this case. You could maybe pause for a few seconds to think about what troubleshooting steps you would take in a case like this. So for us, first step we looked for are just any obvious and easy to rule out factors like reagent lot change. Were there any reagent preparation error, or could there be any random sample handling or preparation errors? And no issue were found on those factors. And then we move to the next step with a revalidation for linearity. Our data shows that our method was linear, and the deviation from linearity was very small across the analytical range. But since then, we still notice in a few batches that high calibrator 5 or 6 deviates from linearity, and it just happens once in a while. And what we did temporarily was just to exclude calibrator 5 and 6 and retest any high samples by dilution. But we wanted to figure out what's actually going on. Then next, we roll out LC overloading or mass spec detector saturation. Lastly, we also considered reagent stability, as the stability of HIA can vary with different pH and temperature. And this seems worth investigating further. So here is what we then assessed for the calibrators under 3 different storage conditions. We tested storing the calibrator in both water and acidic solution, and we also varied the temperature between fridge and freezer, aliquoting or not aliquoting. And after running proficiency tests together with these calibrators, we found that the results stay within the allowable limits across the condition. So it seems like stability wasn't the issue. So, but at least this helped us in ruling out one more possibility. Our next step was to look into other LCMS methods in the literature handling the same compound in the same sample matrix. In our investigation, we included a long list of factors. Some of them are listed here: the sample preparation method, calibrators, internal standards, linear range, also internal standard concentration. Or there are some other factors that's not listed here, including LC conditions, retention time, mass spec instrument, methods, and also the storage condition for everything. Some interesting results actually popped out here. We found that studies that using a deuterated internal standard that has the deuterium at the indole rings of HIAA, we see similar problems reported. And in some papers, they also mentioned potential stability issues of these internal standards. Then we looked more closely at the chemistry of our analyte and internal standard. As we all know, from second-year chemistry, indole rings normally wouldn't allow for a hydrogen exchange. We found out that this older chemistry paper in the 70s showing that when the fifth position is substituted with a hydroxy group and in acidic condition, the 5-position hydroxy group can actually activate the neighboring position 4 and 6 of the indole ring and make them slowly exchangeable protons. So if we have deuterium label at these positions, the hydrogen deuterium exchange may occur in the ionization source or in the gas phase in an acidic environment, which may lead to the internal standard response variability. And this just may be one of the explanation of the linearity issue we have. And then looking back at those methods, for method C, they attributed the positive bias to the stability of the indole ring hydrogen deuterium exchange. But they did not really explain why the internal standard signal drop would cause a positive bias. And for method D, they observe a quadratic fit in the analytical range they reported when they use D5 internal standard. The deviation from linearity was about 16%. They attributed the nonlinear behavior to the retention time difference between the analyte and the internal standard. For method E, they observe a positive bias for some proficiency test samples. They attributed the positive bias to commutability of the calibrators and the QC materials from HPLC method to LCMS method. And last, in the method F, they attributed the nonlinear behavior of different lots of calibrators to the lot-specific problems. And then from all these methods we show here, their explanation of nonlinear behavior are all different, but that one thing is they confirm our issue were not unique, and the chemistry of the internal standard could indeed be a factor in here. So to explain this further, let's look into the chromatograms of the calibrators. I'm showing here as a good day in our calibration. Our cal curve is linear and R square pretty good. On the right-hand side are individual calibrators, and the red arrow are internal standards, and the blue arrows are the analytes. The red dotted lines are the boundary of the peak when the internal standards eluded from LC. Let's look at the analyte boundary in the blue lines. Although there's a slight shift, they overlap pretty well in general. And then let's look at a bad day of calibration, where we see nonlinear cal curve with a R-square of only 0.99. We can see that analyte calibrator 1 to 4 overlaps reasonably well with the internal standard, but at higher concentrations, cal 5 and 6, the overlap breaks down. We can see the analyte elute outside of the time frame of the internal standard, which is highlighted here, which means that the internal standard isn't there to correct for the ion suppression or matrix effect in the ionization process. The lack of correction could be contributing to the nonlinear response we observe. So what would be our solution? We decided to try a different internal standard, a carbon-13 labeled version of the analyte. This one should perform more like the analyte compared to the deuterated internal standards. So when we run the cal curve with the carbon-13 labeled internal standard, we saw some immediate improvement. The R-square of the cal curve is now 0.99996. And if we look at the retention time of the internal standard, they matches exactly with the analyte. And the elution time window is also exactly the same. We then also revalidated the linearity range with this new internal standard and found that the deviation were consistently below 3%, which is well within the acceptable limit, and the adjustment really resolved the calibration inconsistency we see, and this restored our confidence in our assay's accuracy. So what did we learn from this first case? Using a carbon-13 labeled internal standard give us much closer chromatography behavior to the analyte, and this would improve assay accuracy. It is important to remember that the carbon-deuterium bond in a deuterated compound is more acidic than a standard carbon-hydrogen bond, and especially when deuterium label are on the aromatic rings. This acidity can result in an isotope effect where the internal standard elutes earlier than the analyte on a reverse phase column. On a good day here, with a bad LC separation, we actually saw a nearly co-eluting pair. But on a bad day, a slight, even less than 0.1 minute separation between the peaks means that the internal standard wasn't fully correcting for the matrix effect or ion suppression. By switching to the carbon-13 labeled standards, we avoid these very small shifts in retention, which provide a consistent calibration curve. Okay, let's move to our second case, the whole blood immunosuppressant test, which covers 3 different immunosuppressant drugs, tacrolimus, sirolimus, and cyclosporine. This is also a test that has been running in our lab for a very, very long time, nearly 2 decades. And the current sample preparation workflow is shown here. 20 microliter of whole blood was first treated with zinc sulfate and then mixed with 200 microliter of acetonitrile containing internal standard, and then vortex for protein precipitation, and then filtration. The filtrate was then injected into mass spec for analysis. The internal standard of these three analytes are listed here. We use an analog with a slight different side chain for tacrolimus, a CD3 label on the methoxy group for Sirolimus, and a D4 label on the side chain for cyclosporine. We use a 6-point calibrator that contains all three analytes for the quantification, and we also monitor 4-level QCs containing the three analytes. The immunosuppressant is running well for years, but we recently started to notice some imprecision issue for Sirolimus at the lower end. Occasionally, the CV was even worse than the one that's showing here. In contrast, the same QC for tacrolimus or cyclosporine stays within acceptable range for imprecision. While working on this quality improvement project, we decided to update this method using a liquid handler to make the sample preparation process fully automated, which can eliminate human error and minimize random pipetting issues. But the CV at the lower end still stays elevated for Sirolimus. Then we looked back in our original validation data and the earlier QC performance. Although the Sirolimus result CV was generally higher than the other two compounds, they still are in the responsible range, reasonable range. Seems like the current imprecision issue is specific to Sirolimus. And after extensive troubleshooting, we found out that the issue might again be the internal standard. For Sirolimus, we are using an internal standard from vendor A, and this specific lot has a isotope purity of only about 90%, and about 8% of internal standard material is actually D0, which is the same as the analyte. This means each time we add an internal standard, we are adding also a small same amount of analyte into the sample. Here I'm showing our blank sample. We should barely see any Sirolimus signal, but here we see a peak that equals to a signal of about 5 to 10 microliter of Sirolimus. And this will shift our cal curve actually upward a little bit. Our quantification algorithm does include a blank offset where every sample will be background subtracted before plotted on the cal curve. Because of this extra standard addition step, it affects our CV at the lower end. For each measurement, the background can be slightly different, but we subtract the same blank, and we are relying on the accuracy of this one single blank measurement for all the samples across the batch. So even a small consistent error will affect the CV, particularly at low concentration. To resolve this, we switch to another vendor that has an internal standard of Sirolimus, and the isotopic purity is 99.79%. This gives us a 10 times smaller analyte peak in the blank, and we have better CVs at the lower range. So to conclude, we have highlighted the importance of carefully selecting internal standard for LC-MS assays, particularly in clinical applications, where the precision and accuracy of the assay are related to patient care. And In the two troubleshooting cases, we discussed how an internal standard can impact assay performance. In the 5-HIA case, we observe how isotope effect and the deuterium exchange can affect linearity. And for the Sirolimus assay, we learned that isotopic purity can actually affect imprecision, particularly at low concentrations. And going forward, the take-home message is to be mindful of isotope effects and carefully review the data and consider carbon-13 labeled internal standard over a deuterium labeled internal standard if it's available. And by focusing on these details, we can actually continue to improve the performance of our mass spec assays. That's it for today. Thank you for your attention.